Hello everyone, I am Soumya from First MSc Microbiology. I am going to be explain the topic of the complement system. Complement system is the part of immune system. Complement system that enhance the ability of antibodies and phagocytic cells to clear microbes. It is the part of innate immune system. The term lysine was renamed as complement, which was coined by the Paul Ehrlich. The complement system consisting of over 30 proteins. It was produced by the livers and found circulating in the blood, serum, within tissues throughout the body. The complement system was first discovered by the Jules Borat in 1895. He established that human bacteriolysis and hemolysis requires two factors: heat stable antibodies, heat labile components. The immunoglobulins of IgG helps the immune system to trigger the complement system. The complement does not bind with free antigen or free antibodies. It can only bind with the antigen-antibody complex. Complement system, how it is activated. First, antigen-antibody reactions called classical pathway. Second, bacterial endotoxins called alternative pathway. Third, lectin pathway called mannose binding lectin. Components of complement system. Recognition unit C1. C1 contains Q, R and S. C1 Q's functions of binds to Fc portion of antigen antibody complex. C1 R functions are activate C1S. C1S functions or cleaves C4 and C2 due to its enzymatic activities. Second, activation unit C2. C2 functions causes viral neutralization. C3 are A, P and E. C3A contains anaphylotoxin. C3B are key components of the alternate pathway. C3R induces leukocytes. Third, membrane attack unit. C5 components of A and B. C6, C7, C8, C9 functions of participate with C5B in formation of the membrane attack complex that lyses targeted cells. Alternative pathways of factor B, factor D and properdin. The regulatory proteins contains factor H, factor I, C4b binding protein, C1NH complex and S protein. Functions of the complement system. Cytolysis or cell lysis. The first function of complement is inducing the cell death of membrane attack complex into the targeted cell membranes. The components of membrane attack complexes are C5b, C6, C7, C8 and multiple copies of C9 where rupture the microbial cell surface which kills the cell. Opsonization or phagocytosis. C3b bonds to the immune response complex are coated on the microbial cell surface which kills the cell. Opsonization or phagocytosis. C3b bonds to the immune response complex are coated on the surface of the pathogens activate phagocytic cells. These proteins bind to specific receptors on the phagocytic cells to get engulfed. Inflammation. Smaller fragments of C3, C4 and C5 cleavages of C3A, C4A and C5A act as the inducer of inflammation. All the three inducers can bind to the receptor on the mast cells and basophils, causing the degranulation and release of histamines. Histamines induce smooth muscle contraction and increased vascular permeability. Combination of C5A actions on mast cells, endothelial cells and neutrophil contributes to the process of inflammation. Complement clears immune complexes from circulation. In immune complex activates complement and C3b is generated. C3b coats the immune complexes and CR1 on erythrocytes bind C3b. The red blood cells bind these immune complexes and take them to liver and spleen. Phagocytic cells removes immune complexes from the red blood cells. Enhancement of antibody productions. Bindings of C3b to its receptor on the surface of the activated B cells enhance the antibody productions. Chemotaxis. Movement of cells towards food and away from toxic materials. Some other functions of complement systems are neutralization of viral infection, induction of immune response, disposal of hepatotic cells and bodies, activation of leukocytes, lysis of bacterial cell wall and result in the apoptosis or cell death. References: QB Immunology 6th edition, Immunology Nandini Shetty 2nd edition. Thank you.